A poll.
Oh yeah, I don't think any polling.
Hello everyone. So let's keep going with the emergent sessions. We have Truong on the screen. He will be talking us a little bit. So Truong, I'll stay in the background. If you need anything, just let me know.
Okay, thank you.
Okay, thank you. Okay, thank you. Okay, thank you.
I'm hearing some feedback.
This one comes from any of you, sorry.
All right, let's try this again. Yeah, okay, I don't see hear the feedback anymore.
Yeah, okay, I don't see hear the feedback anymore. Yeah, okay, I don't see hear the feedback anymore. Yeah, okay, I don't see hear the feedback anymore. Yeah, okay, I don't see hear the feedback anymore.
I'm still receiving your feedback. Not sure why. Can we take this offline and then
So, hi, Kay. Perhaps do you want to start? Do you want to start talking about?
I can start. I don't have the slides handy. Let's see. Hold on. I can get them out. Just a second. And I'll let Young take over when he gets into this. So let's see, OSR. Okay. So I'm going to share my screen and go ahead and give the talk until Young gets situated, since he was going to give the talk. Okay, so this is a talk about how to annotate events in time series using HED. And the objective is to get rid of jargon and keywords and to be able to do cross-study analysis with data that is being shared. And this
hi, Kay.
Hi, are you back?
Okay, all right.
Okay.
I think I'm very sorry for that.
Yeah, okay. Young is our designated speaker here. I'm going to stop sharing and let him go to town.
Sorry, everyone. Let me start sharing my screen. Can everybody see the presentation mode? Okay, then I guess I can start. So yeah, I believe Kay started introducing our topic today. So we will be talking about the Hierarchical Event Descriptor project that me working with Dr. Scott McCaig at UCSD and Kay at University of Texas San Antonio is working on. And our goal is to provide a common framework to annotate the nature of events in time series data, that we can hopefully move beyond the practice of using jargon keywords and trigger number value and into the realm of fine-grained analysis-ready annotation for neuroimaging data. So with open science and more data being shared, the need for having common standards to describe data so that user can understand and make use of the shared data is more important. And specifically for high-resolution time series data that measure the electromagnetic activities in the brain, so for example, EEG or MEG, event-related analysis are the central focus of the research interest. So we really pay attention to how event information are documented and described in the experiment. And the BIT standard provide best practices for how we can organize and specify various level of information about the data and metadata, but it still left open the issue of how to tackle the standardization of event description for the experiment. So there is a gap between the data being shared and how it can actually be analysis ready. And when we say analysis ready, we mean that we really want to have a detailed and unambiguous description of the event in a format that it can be acted upon by analysis software. We not only want to document what happened during the experiment, but we also want to document other aspects of the experiment design. For example, the condition or control variables, the task, if possible, the cognitive processes that are undergoing throughout the recording. Another aspect, and this is particularly relevant for high-resolution time series data, is that we know that the brain meets the challenge at every single moment. So if possible, we want to have a way to analyze the dynamic of the brain in its naturalistic environment. So for each event occurs, we want to be able to analyze the event based on the context information in which it unfolds. And so the current practice is that we usually have a set of event code in the experiment that is specific to that recording or to the lab. And these event code documentation is oftentimes, the detail of these event code is oftentimes not documented much later, for example, during the publication process. So by that time, a lot of details about these events are already forgotten by even the researcher itself. And so if we go further, 6 months later, a lot of the nitty-gritty details about the experiment might get lost from any of the documentation. Now, the problem is even worse for people who inherit your data and try to make sense of it, because now they have to dig into the publication itself to find information about the event. And it happens very often that researchers have to email the data authors to ask further clarification question or ask for more information that relevant for their analysis, but not provided by the data author just simply because it was not relevant for the analysis performed by the data author. And the last point is that the practice of having event code in the encrypted code as in right now is that you have to write custom code every time you want to extract information for your scientific questions. And so we really want to have a way to describe and organize information about event in a way that include all the different aspects of experimental event, and in the way that is both human readable and machine actionable. And so we believe that the hierarchical event descriptors framework, in combination with bits and support from analysis software like EEG Lab, will provide a practical path forward to solving these issues. So we will use an example experiment to demonstrate our points. And so the experiment we will be using is the Sternberg memory task. And this experiment was performed by Julie Anton when she was a researcher at the Schwartz Center at UCSD. And the task is to investigate the EEG dynamic during short-term memory task. And so for this experiment, a trial starts with a 5-second display of a fixation cross. After that, there will be a sequence of A letters. Some will be in green color to indicate that they are the target letter to be memorized, and some will be in black color to indicate that they are non-target letter to be ignored. Follow the letter sequence will be a dash for a 2 to 4 second period. This indicates that the subject can try to memorize the letter was shown. And after the dash disappears, a red letter will appear on the screen, asking the subject to indicate whether this letter has appeared in one of the target letter before or it was not. And so the subject will press a right mouse click to indicate that, yes, this was a target letter, or a left click to say that, no, this is not the target. After the subject responds, then a auditory feedback will be presented. It will be a beep if it's correct, or a buzz if it's incorrect. And the subject can press any of the button to indicate that he or she is ready to move on to the next trial. And so there actually more happens for this experiment than that, because in some of the recording, the letter will be presented for 1.2 seconds. But in other set of recording, the letter was presented for much shorter durations. And subjects also was asked to perform additional tasks of alternating between eye open and eye closed for 2-minute intervals. And none of the information about the condition and the task was readily available from the data itself. So again, data user will have to go into the publication of the paper and other auxiliary file to pull out those information. And the way that the event was recorded, and here we show in the EG lab format, but the design of listing event in an event table is common across other analysis software as well as the data standard. So each of the event table, each row of the event table is its event, and there are 2 columns that are universally recognizable by EG lab, which is the latency and the type. So latency is the number of samples of this event. It's just a side note. In other framework, latency can be millisecond onset from the beginning of the recording. The type column contain all the different event codes for these events. And so oftentimes, researcher will try to come up with a good name to encode the event using these event types. And potentially you can also include other columns to describe your event, but those columns are custom columns, so they're not easily recognizable by the software. So you have to write extra code so that the software can filter them out and understand them. So I will go over quickly what these code means that you see in the type column here. So for our experiment, each of the letter presentation have their own event with their letter value. If they are the target letter, then there will be a G prefix to say the green letter. The red letter will be prefixed with an R for probe letter, and then black letter will be simply just the letter value. We also have non-WM and WM to indicate whether it's a non-working memory period, so that is the display of the fixation cross, or WM is the working memory period in which you have the dash appear on the screen. And then we have the button press event, so coded by numerical values. So 1 here is to indicate it's a right click, and will be another value for the left click. And then we have correct or incorrect event to encode the feedback events. So problem with this, and there are various problems with this encoding, is that, so first of all, just by looking at these event type, it is not apparently clear what they mean without having referred to the documentation. Second of all, every time you ask a scientific question and you want to be able to extract events related to your question, so for example, if you want to ask what happened in the brain during the target letter presentation, then you have to subselect a set of the event code, and here we have will be 26 different event code for each of the letter value to answer your question. And so every time you have a new question, then you have to figure out a way to subselect and filter the event of interest. Now, another problem is that if you notice the button press event, both of the button press here have the same code, but they mean different things with respect to the experiment. So the first press means to indicate a response for the probe letter, but the second press is just simply means that they are ready to move forward to the next trial. And if in order to parse out, if just we interested in the button press event with respect to the stimulus, then we have to write some code to parse out the specific event based on their when they appear in the recording, and those could be a non-trivial task. So we will show how HED can provide a path forward to transform this practice into a more human readable and also readily actionable form from analysis software. So first, we want to show how we can rethink about the way we design the event table with respect to the HED annotation. So again, the current practice use only one column to encode every single possible information about these different event types. But with using HED, now we can design our event table in terms of the orthogonal components with respect to the experiment design. So in most cases, we want to separate out what happened during the experiment, so the type of the sensory and action, and the purpose of those events. So we have many different purposes for them. So you can see how we move from having 84 different event codes to encode every single possible thing that happened during the experiment into 7 different types and 10 purposes with respect to the experiment design. And we also have a letter column here to contain the specific value of the letter that was shown on the screen. And we will show next how, given this orthogonal event design, then we can very easily annotate what happened during the experiment using HED. So here an example of the HED annotation for the event design that's shown previously. And so, for example, the left-hand side is the table annotating the type column. So each of the row is the unique level value for the column type. And you can see how already the HED annotation associated with this type is meaningful on its own. So we can see that we have a sensory presentation event. It was a visual presentation. It contained a shoulder cross. And similarly, we can annotate the purpose column using HED tags. And then for the numerical, the value column, the letter, so we have many different letters. With HED, we don't have to annotate a HED tag for individual letter value. We can simply specify one HED annotation, including a hashtag symbol. And then during the analysis time, the HED software will automatically assemble replace this hashtag with the value of the letter column for you. So for example, here, let's say we want to assemble the HED annotation for this particular event of show letter as a non-target show letter with the letter D. You see how below the HED software automatically assemble the HED tags associated with each of these column value, concatenate them, replace the hash symbol with the letter value, and provide you a complete annotation for this specific event instance. Now, given this event annotation, then you can go on and start asking some question simply by specifying the HED tags. So, for example, let's say we want to ask what happened for the non-target experimental stimulus presentation. We can just simply say specify experimental stimulus and non-target in our HED query. And I will show you a specific demo later on. You'll soon see how it worked out in each lab. So the process I just described in terms of the EG lab environment, also applicable for BITS. So HED had been accepted by BITS for a while now. And so in BITS, the event table is stored in the events.tsv file. So it's similar to the event table in the EG lab event structure. The HED annotation can be stored in the events.json file. And with the inheritance principle of bits, one thing we really want to make note of is that you can simply have one single events.json file at the top level of the bits data set, and the HED annotation can be applied across all of the different recording within that data set. And this is similar to the study mechanism of EG lab, in which you can have one single annotation for the study level, and that annotation can be applied across the EG lab data set. So given what I have previewed about the benefit of annotating and working on your event analysis using HED, I'll go into more details about HED as a standard itself. So the essential component of HED is the HED schema. It is the control vocabulary organized in a hierarchical manner. And so, for example, on the left-hand side here, you see the HED schema, stable view. So we have 8 different subtrees in the HED schema. The event subtree will show different category of the event. We have action to describe action in the experiment, item containing different things like objects that appears in the experiment. And then we have other properties and attributes subtrees that you can use as modifiers for the items above. A head task, as I showed an example before, is simply a path in the schema. And your head string can contain multiple individual tags, or the task can also be combined using parentheses. And for example, if you want to annotate a green square, for example, then you can say green and square in parentheses together to indicate that the green is a modifier of the square. Again, as I mentioned, the HED has been accepted under bit standard, and so you can have a HED annotated data set under bits. And we believe that some features of the third generation of HED, so we call them HED 3G, have significantly improved the capability and usability of the HED system for users. And I will show details next. So one of the improvement in the third generation of HED is the idea of unique mapping. And so in this concept, each node in the schema, so for example, sensory event, must be unique across the entire schema. And given that, then you can have user use just the node name on its own instead of the full path to the node for their annotation. So for example, they can just write cross instead of having to say item / all the way to 2D shape and cross. And you can see how the short form head string using the unique mapping node name is much more readable and understandable comparing to its equivalent long form. Now you might ask, why do we want a long form at the first place? Well, the long form is valuable, especially when you want to do cross-study analysis. So you might not be interested specifically in what happened during green visual presentation or red visual presentation, but you might ask questions such as, what about a color visual presentation in general? So with the expanded long form, then you can ask more general question, and the HED tool will be able to detect a more common and general annotation from the event. I'll show next a quick demo of how you can use EG lab to annotate the data set and perform some analysis based on the annotated data set. So similarly to any of the HED plugin, you can install using the HED extension. So the plugin name is the HED tools. And then once you have the plugin installed, you can load a dataset, HED dataset, for example. And here is the event structure. So we have designed the event structure to be similar to the event design that we recommend with orthogonal components. And so once we have our dataset loaded, we can go to edit event head tags. And then it will ask you to specify the categorical fields. And because if you remember earlier, the way we annotate categorical column is different from the way we annotate the value column. Then the C tagger, our GUI interface, will show up. The first thing is that the top panel will have a drop-down menu, like different event field, pull from the HED structure itself. If you select a categorical field, then it will list all the different unique level of that field on the left-hand side panel. But if you select a value column, so for example here duration, there will be one single item called head, in which you can associate your single head string with the hashtag value with it. And then to start your annotation, you can click on the button Show Schema, and it will bring up the head schema in an expandable view. So you can collapse and expand and navigate to explore different head node that might match your annotation intent. So clicking on one of the node will bring up the description of the head schema node. And then if that matches your annotation intent, then you can double click on it and it will be automatically added to the editor panel on the right. And so the process of annotating with your data set with head can simply mean browsing the head schema and choose different head tags that match your information that you want to describe about the event field level. You can also directly type into the editor, and as you type, C tagger will look up different tags that match your partial keyword and show the suggestion for you. And so when you choose one of the search results, it will automatically replace that with your partial key enter. And so during the annotation process, then you can review your progress so far. And you can also save your progress into events.json file. And this is helpful if you want to share your annotation with other people in the lab. Or also, you can import annotation that prepared by other people in your own data using the import function of C tagger. And so once you finish your annotation, then you can see a final view of all different annotation that you have. And then you can continue, and each lab will ask you to whether you want to create a new data set. And so this will be a tagged version of your data set. So once you tag a data set, and the mapping of the event field and the field level with head tags will be saved into a structure in the HED lab. And I also show an example here of how the head tools automatically assemble the head annotation given what we have specified in C tagger for each of the event. And given this annotation, you can now use the epoching tools by head tags to start asking scientific questions. So when you select the menu to extract epochs by tag, a window will show up. And on this right three-dot button here, it will bring up a user interface for you to search for all the available tags in the data set to form your head query. And so, for example, let's say I'm interested in seeing the process for target sensory event. Then I can just provide this head query into the epoch head windows. It will create an epoch data set. And now I have my epoch data set based on the target presentation. So you can quickly plot the average ERP image, we call ERP image in HED lab, and show the ERP for that particular target event. And if you have a different scientific questions, we can simply use another head query on the tag data set. For example, here I want to ask what happened for the non-target, or also, let's say if I'm interested in analyzing the button press event. So with that, I hope that I have shown you how simple it can be to annotate your data set given a head-supported tool, and also how performing analysis using head tag is more meaningful and also easier comparing to having to deal with the cryptic lab event code. So with the third generation of head, we have introduced many other new features. One of them is the library schemas. So one problem we observed with using the previous version of head is that the schema can be proliferated very easily with term only specific to a specific subfield that might not be relevant to the general purpose. And so we introduced the concept library schema. So now sub-community can develop their own schema, and during annotation, they can use their library schema in combination with the base schema for annotation. So we already have a clinical EEG library schema in development, SCORE, and we plan to also develop a video event library schema for annotating experiment involving movies. Just one caveat is that the validator for validating library schema in BIS is not being fully implemented yet, but it will be ready soon. One other question we often get asked when we introduce HED is that whether HED is a formal ontology or can it be used in combination with an ontology. And so HED is not on its own a formal ontology, but it's meant to complement a formal ontology. So we want HED to be user-facing technology, meaning that we want it to be straightforward and easy for user to start using HED and annotate the data with HED. In comparison to formal ontologies, we usually take a lot of learning curve to get used to work with the system. HED also designed to be able to document various other aspects of the experimental event that formal ontology might not be able to express, for example, the temporal scope of the task. And so, but that being said, HED also have a mechanism so that you can associate the ontology ID that match a node in the head schema, and that way you can establish a link between the head schema vocabulary to the formal ontologies, and you can make use of that as desired or needed. And we also have specific tag under head schema, so you can provide the cognitive atlas or CogPO ontology ID. Another features we introduced in HED 3G is the definitions. And so definition in a way that you define a variable in your programming code. And with definition, you can associate a custom name. So for example, some lab-specific terms, and you associate that term with some other common head tags pulled from the head schema. And in this way, you can use this definition name during annotation in place of the long head tags associated with it. And now the definition mechanism also enable us to specify the temporal scope of the event. So if you can associate the definition name with the onset and the offset, then you can specify the temporal scope of a particular event type. And more importantly, the ability to specify temporal scope now enable us the possibility of analyze context-dependent event analysis. Oh, and quick note is that during analysis, the head tools will automatically expand the definition by its associated tag for you, so that all the tag associated with the definition will be readily available for epoch tool or analysis tools based on head tags. So we have developed several software to support the head standard. The validators are being maintained in both Python and JavaScript to be able to work on the web and also under Bits Validator. I have shown you the demo for the EEG Lab plugin for HED. where it assists you in the annotation process and also start analyze your EG lab data using HED tools. The HED schema, the official HED 3G schema, will be available and published very soon. And we also plan to work with Data Lab, which is a data management software. I believe that a lot of you here know about it, because Data Lab is the backbone of many data portal, including OpenNeuro and Sumi Mimar. So we want to be able to have HED information understandable by Data Lab metadata format, so that any tools using Data Lab to manage their data can start doing event discovery using event HED annotation. So as with any standard, the value of the standard is only actualized when people start using it. So we really hope that the community will start using HED and you start annotating the data set with HED. So if you have interest and want to start the process, please do not hesitate to contact us. You can also check out the HED official webpage, headtax.org, for more information. We'll be providing more documentation and tutorials to come. And we also have an extensive case study paper available detailing specifically how you can use HED to document various aspects of experimental events. We want to thank our collaborators and also our funding sources. So now we want to open it up for any question and comment.
Thank you, Young, for your presentation. Very nice.
Thank you.
You have mentioned at the very beginning that there is a possibility to integrate all of this within different toolboxes or software, right? But you have tested till now only on EEG Lab?
Right, yes. So we would definitely love to have other toolbox and another software to start supporting HED. But for now, since especially our project started from the SCCM, which is where the EG Lab toolbox come from. So we start our development with putting it into EG Lab. But yes, definitely.
Makes sense.
Yep.
But how easy do you think will be the integration into different toolboxes?
Yeah, so Kay can give a detailed explanation on this, I believe. But the HED schema is provided in an XML file. And so, the first step is simply parse the XML file so that you can get the HED schema in there. And also, we have all the online tools that can be made into server that people can request for validator or assembling of the HED annotation. So, yeah, Kay, you want to?
Yeah, let me say a few more words about that. So we have a Python library that everything is open source, and the web tools run in a Docker module, but separate from that is a Python library, a HED tools library, and that will do validation, it will do assembly, and a number of other operations. And we plan to do additional tools that would be callable in Python. All of the tools that are in Python are callable through the web tools by a web service. So tools can also do that. And we've been talking to some other tools, but until we, once HED 3G is released and we have our basic tool set completed, then I don't see a problem in having it be integrated into other tools. And it's all open.
That's great. That's really great. And how easy will it be to have it for different vendors? Because I mean, you highly rely on how the specific toolbox handles the data from an EEG system, right? So if you are using, I don't know, I can just think on our own, in which we do not always use the standard system, like a CAP system. Sometimes we just want a few electrodes, and we use a different box or a CDE or some other system. How easy will be that for us to implement?
Well, it depends on how you express your events. You know, if you're using a spreadsheet model of your events, it will be very easy to integrate. If you have some other model, they almost always map into a spreadsheet model. You know, I mean, when you get down to it, regardless of how many electrodes you have. You know, it's very bids oriented. So and a lot of the tools are now going to supporting bids, at least for their basic low level integration of raw data. The derivatives, that's less well developed. And I think, you know, there's a community working on derivatives, and whatever they do will fit in.
Well, let's hope so.
But we haven't gotten that far. Yeah, I mean, they haven't gotten that far. So basically, we're thinking about events in terms of spreadsheet models. You have any number of columns. You can call them anything you want. You can have any number of things in your columns. And it'll work out of the box right now. Things that won't work are if you have columns that have complicated data in them, like lists of things. It assumes that each column has single values in the columns. So, you know, if there were a good reason to do lists, we would do lists. But right now, you know, I think we need to focus on getting basic tools and also some downstream analysis tools that really leverage this. And one of the things that that Young hasn't mentioned is that it does, the whole mechanism allows you to do control variables and experimental conditions. And so you can extract those automatically. And so you could do automated tools where you want to find all the stimulus under under different experimental conditions and just say, I don't care what they are, just, you know, look at them and compare them and look at whether there are any statistical differences. That's something that could be automated now, I mean, very easily. Another thing that's really important, it is being able to do temporal overlap regression. And the model that we use for the events really encourages or enables you to, you know, implement tools that will regress out at least the linear interaction of different events. And we found that's really critical, especially if you do cross-study comparisons, to regress out things, you know, the different overlapping things, because every experiment has got a sort of a different lineup of the overlapping events. This is very powerful from the viewpoint of reproducible science, you know, being able to see whether it was just your specific way of setting up this experiment, or whether this is something that's really generalizable. And that's really why we're pushing it, is because we see an opportunity to do that.
Right.
That's true. And is there any quality check on the data itself?
Can you amplify that?
Yeah.
So what can you by quality?
Yes, either like proving that namings and everything is correctly assigned, for example. I mean, I guess it's also related to the derivatives, which you already mentioned. It's not fully implemented.
I'm still not quite following. Can you kind of?
So let's say it a different way. What are the things that might be wrong while using your tool?
Well, so it all depends on how well you annotate. And one of the things, so we've actually done some large-scale studies across experiments and labs using HED version 2, which was not nearly as capable. And what we found was that we sometimes had to go back, and different people annotate differently. And when you want to do cross-study comparisons, sometimes you know, even though they're talking about something that could be compared, the annotations, your analysis is only as good as the clarity of your annotations. Right. And we ended up having, you know, some labs did better than others at doing the annotation. We ended up having to re-annotate. And one of the key things about this is that for the most part, you just need, you have the annotations separate from the actual data. They're sitting in these dictionaries. And if in most experiments, you only have to have one dictionary. And that makes it easy to adjust how you're going to annotate the individual events, so that if you can sort of fix annotations fairly easily and, you know, do a reanalysis.
So do you have any kind of guide or recommendations for doing annotations?
Yeah, well, we have this case study, and it's about really a best practices, not only in annotation, but a lot of it is event design and what you report for events. And what we found is that when researchers take their data, they tend to think about that one paper that they're going to publish, you know, when they get this experiment done, and not about what this data is capable of being used for. And so they only report the bare minimum of events, the ones that they're interested in. And it turns out that they leave out all of the secondary stimuli. So the subject is sitting there and they're seeing the fixation cross, and they're seeing, you know, the cue, and they're hearing a buzzer. None of that is in the event thing. Right. You know, they just have stimulus. Yeah. And the problem with that is, if you want to regress out the interference of these other things, especially if you're doing like visual stimuli and you have visual cues and stuff, there's going to be interference. And you've already, if you don't report all your events, you've already, you know, eliminated the possibility of using your data for that kind of analysis. So we have this case study where we're really trying to promote a good strategy for how you report events and what events you report. And that's probably more important than the details of how you tag it, because you can always go back and adjust the tags. But once you don't report the events, you're gone. So that's the message. Report your events and report them in a reasonable way.
Yeah,
that's pretty cool. Perhaps you can also add the link in the chat so that it's available for people. And we have also one question from Ramon Martinez Cancino. He's asking whether the slides are available somewhere.
Yes. Do we have the slides somewhere? Yang, you want to put them up somewhere?
Yeah, perhaps I also made a reminder that this video will be put on YouTube, sorry, so everything will be available afterwards.
We'll put it on www.headtags.org. And no, Alpha 2 is not the final release. We're currently on beta, but it's not up yet. And the final release will have no alphas or betas. It will have 8.0.0. But we're working really hard to get that final release out. It won't, the vocabulary won't change so much, but we've revised the schema format somewhat so that we could use XSD, a schema validation, as to make things, and there are some technical things that we're still trying to do, and we're trying to make sure the tools can read all the formats before we release. But there won't be much change there's some minor changes in vocabulary between alpha 2 and beta, and I don't anticipate any changes between beta and 8.0 as far as vocabulary. It's mainly our getting the tools and the underlying format, making sure that it's all consistent.
Right.
Well, that's great. So it sounds like still a lot of job, a lot of work to do.
Yes.
So if you have any further comments.
So let me say one thing. If you're a data provider out there, someone who has an experiment, and you want some help or suggestions in how to report your events or how to annotate your data, email us. We're going to be doing a lot of annotation this summer to try to get a collection of example data sets from a variety of fields. And, we would be happy to make suggestions and try to be helpful and hopefully make your data more usable and visible to the community.
Right.
Right. Okay. Then I have to thank you both for the.
I think there's a glitch.
Yeah.
Okay. Well,
okay. Well, we're going to say, and thank you all for listening.
Yep. Bye, everyone.
Bye.